Hello everybody. Today our topic is Mendel's laws of inheritance, and this topic will be completed in three parts. The part first, which is the current lecture, will include details about all the experiments that Mendel carried out and how he formulated the laws of inheritance. This lecture will also include the law of paired characters and the law of dominance. Then part second will include the law of segregation, and part 3 will include the law of independent assortment. So first we need to know about the Gregor John Mendel. Gregor John Mendel was born in a very poor family, in a peasant family in 1822, and he had to struggle a lot during his studies. And from 1856 to 1865 he carried out several experiments on garden peas and formulated the laws of inheritance, and he published his findings he published his findings in 1866 in Annual Proceedings of Natural History Society of Bern. It's a journal, and he published his findings in this journal. His laws form basis for the science and genetics even today. As such, he is remembered as the father of genetics because his laws are so much important. They even today form basis for science and genetics. The law of segregation and law of independent assortment. As such, he is remembered as the father of genetics. But unfortunately, Mendel's discovery was not appreciated at that time by the scientists, by the biologists, by the contemporary biologists because of several reasons. First was his work was ahead of his time. The research that he carried out was ahead of his time. It was difficult to understand at that time because science, there were very little advancements were made in the field of science. Nobody had knowledge of genes at that time. There was no concept of alleles at that time, so his work was ahead of his time. He second is he published in a journal which had very narrow, which had very limited readership. Very few people actually read this Annual Proceedings of Natural History Society of Bern, this journal at that time. So it has very limited readership. Then third, he was himself not sure of his findings. So when he was not himself sure of his findings, he did not struggle to discuss it with the contemporary biologists at that time. Even he had tried certain times, but he failed. And then fourth was that he lacked aggressiveness. He should have been aggressive in nature to discuss these findings with the fellow scientists of at that time, contemporary biologists at that time, but he failed that in doing so. And fifth is his mathematical approach in working with biological problems was strange. The scientists at that time, they used a different approach, while what Mendel used at that time was strange for the scientists. So it was difficult for them to understand. And the most important was that at that time Darwin had proposed his theory of evolution, and every scientist was preoccupied with that Darwin's theory of evolution. And so the Mendel's laws of inheritance, Mendel's findings, they received very little attention. And finally, Mendel died of kidney disorder in 1884 without knowing that he had actually laid foundations for modern genetics. It was not until 1900 that three scientists, Hugo de Vries, Carl Corns, H. von Schmuck, they carried out hybridization experiments, and they rediscovered Mendel's findings. Now, in 1900, Hugo de Vries of Netherlands, Karl Corns of Germany, and H. von Schmuck of Austria, they independently carried out hybridization experiments, and what they found was what Mendel had already reported in the journal, which had very limited readership. which we saw in the earlier slide. So they rediscovered the Mendel's laws. They found that Mendel's findings, they were actually true, and they gave all the credit to Mendel. They were very honest, so they gave all the credit to Mendel. That's why Mendel is today known as the father of genetics. And Mendel's work was republished by De Vries in 1901, and later on Bateson and many other biologists, they worked on hybridization experiments and others, and they found that same laws also apply to animals. So these three scientists, they rediscovered the Mendel's findings. First was Hugo de Vries, second was Karl Korns, third was Erich von Schimach of Austria. And now, why pea plants? Why Mendel selected pea plants for experimentation? Now there were certain specific advantages of selecting pea plants. First was the contrasting traits. Now what's a contrasting trait? Any trait which appears or which always appears in two opposing conditions. Any trait any trait which any trait which always which always appears appears in in two opposing conditions opposing conditions two opposing conditions. Any trait which always appears in two opposing conditions is known as contrasting trait. Now we have different traits in in pea plants. For example, we have seed shape, we have seed color, we have flower color. These are different traits. Now seed shape appears as two opposing conditions. In case of seed shape, we have two conditions. Either the seeds may be round or they may be wrinkled. So these traits, they can be used as markers for carrying out the experiments. They can be used as markers. By observing these traits, you can say what kind of a plant this is, and what kind of plant this is, what kind of a genotype this plant may have, and so on and so forth. So Mendel selected pea plant first because of the this pea plant. It exhibited some contrasting characters like seed shape, seed color, flower color, and they were actually 7 contrasting pairs of characters contrasting these traits. Now second is self-pollination. The plants can be easily self-pollinated. The flowers are bisexual. As such, they self-pollinate, and which is very important for maintenance of the purity of the characters. Third is the hybridization. You can easily carry out hybridization experiments on such plants. The anthers are large, you can remove them, emasculate them, and then you can carry out the hybridization experiments. Then third point is the lifespan. The lifespan of the pea plants is very short. They complete their life cycle in a short period of time, so you can carry out many experiments on them. If you carry out experiments on a plant who completes its life cycle in years, then it will be very difficult to repeat the experiments, analyze the results again. So when a plant has a small lifespan, you can carry out experiments in replicates. You can have several repetitions of your experiments to confirm your results. Then seed number. The seed number in case of pea plants is large. but it's not that large. It is appropriate for calculations and all formulation and etc. So seed number was also appropriate for carrying out the experiments. The pure lines can easily be generated in pea plants because you can self-pollinate them. So you can generate the pure lines in case of the pea plants. The seventh is availability. You can get the seeds from anywhere. They are easily available and you can get them and perform the experiments. Now we move to Mendel's laws of inheritance. Based on his experiments, based on the experiments that Mendel carried out on the pea plants, he put forward different laws of inheritance. Now, first two laws, the law of paired factors and the law of dominance, these two laws, they are not universally acceptable, they are not universally true. They apply at some instance and at other instance they do not apply. And the two main laws, the first law, the law of segregation, and second law, the law of independent assortment, they are universally acceptable and they are the main laws of inheritance. Now, to understand these law of segregation and independent assortment, we also need to understand the law of paired factors and the law of dominance. First, we'll look at the law of paired factors. Now, the law of paired factors, it states that a hereditary character is controlled in a diploid organism by at least two alleles that occupy the corresponding loci in the two homologous chromosomes. Now, three important terms have been used in this statement. We need to know all these three terms. First is the hereditary character. What is a hereditary character? Yes, you are right. A hereditary character is a character which can be transferred from one generation to the another generation, or from parents to the offsprings, or from one generation to successive generations. For example, the color of your hair, for example, your height, for example, the color of your eyes, the color of your skin, all these, they can be transferred from one generation to another generation. As such, they are the hereditary characters. Second is homologous chromosomes. Now, what are homologous chromosomes? For example, we have 46 chromosomes. All these chromosomes, out of these 46 chromosomes, we have 23 chromosomes that we receive from mother, and 23 are we receive from the father. So 23 are paternal and 23 are maternal. So overall we have 23 pairs of chromosomes. Each pair represents a homologous uh, uh, chromosomes. Each pair is a pair of homologous chromosomes. So in each pair we have one chromosome from mother and another chromosome from father. The two homologous chromosomes, they have same length, they have same size, the number of genes is same, the position of the centromere is same, the bending pattern, the standing pattern is same, and on each of these chromosomes we have genes. Now, according to the law of paired factors, if we have a particular character, for that particular character, we will have a gene on this chromosome, on a gene on the chromosome which is we inherited from the mother, and we will have a gene on the chromosome which we inherited from father. So we'll have one copy of the gene on the maternal chromosome. At the same position on the chromosome that we received from the father, we'll have another copy of the gene. Now these two copies, these two alternate forms of the same gene which are present on the maternal chromosome and the paternal chromosome in a pair of homologous chromosomes is known as the allele. So allele is the alternate form of a particular gene. So in a homologous chromosome we have two chromosomes, one is maternal, one is paternal, and we have genes on both these chromosomes. The genes are same, they are similar on both the two chromosomes. Each gene has one copy on the maternal and another on the paternal. These two alternate forms are the alleles. Now these two alleles, for example, we have two chromosomes, one is maternal and one is paternal. For a particular character we will have a gene we will have an allele on this chromosome and allele on this chromosome, an allele on the maternal chromosome and allele on the paternal chromosome. So these two alleles, they will actually control a particular character. This is what law of paired factors says. Or we can say that an individual receives one allele for a particular trait from each parent and passes one of the two alleles for a trait at random to each offspring. The two alleles for a trait are when the two alleles for a trait are similar, they are known as homozygous, and the individual is known as homozygous individual, and when they are dissimilar, it is known as heterozygous individual. For example, I will give you an example here. What is homozygous and heterozygous? Now, in case of plants, we have a gene T. It is responsible for the height of the plant. Now this gene has two alleles, small t and capital T. Capital T, this allele, if this allele is present, the plant will be tall, and if we have small t, the plant will be dwarf. Now we can have three types of individuals. We can have three, since we receive one allele from mother, one allele from father. We can have three types of individuals: individual 1, individual 2, or plant 1, or plant 2, and plant 3. In case of plant 1, the plant receives capital T from father and capital T from mother. Individual 2 receives small t from father and small t from mother, and plant 3 receives capital T from father and small t from mother. Okay, so we have three individuals. In this plant, both the alleles are similar, then this individual is known as homozygous. In this case, both the alleles are similar, so this allele is also known as homozygous individual. And in this case, you have two dissimilar alleles. Although both are responsible for a particular character, both the alleles are responsible for the height of the plant, but they are not similar, they are dissimilar. Such an individual is known as heterozygous individual. So for a particular character, for a particular trait, we have two alleles. One we receive from mother and one we receive from father. So this is what law of paired character states. Law of paired character states that for each character, for each trait, we have two alleles. We have two alternate forms of gene, and one we receive from father and one we receive from mother. And now if both the alleles, they are similar, for example capital T and capital T, small t and small t, when they are similar, it is known as homozygous condition. When they are different, capital T and small t, it is known as heterozygous. Now we have the law of dominance. Now the law of dominance states that one allele of pair of alleles which controls a character usually expresses itself and prevents the expression of the other. The allele which is expressed is called dominant allele, and the unexpressed one is called a recessive allele. Now in the law of paired characters, we understood that each character in an individual in a plant is coded by two alleles, and according to law of dominance, out of these two alleles, one is expressed and another is not expressed. The one which is expressed is known as dominant, and the one which is not expressed is known as recessive. I'll make you understand with the help of a simple example. For example, uh, in case of plants, we have an allele T for tallness and allele small t for dwarfness. For example, we have two individuals or two plants, one plant has capital T and capital T. Because capital T is responsible for the tallness, the plant will be tall. The plant will be tall. Another plant has small t and small t. Because small t is responsible for the dwarfness, so the plant will be short or dwarf. Plant will be dwarf. Now, during meiosis, here you will have two gametes. You will also have two gametes over here. Okay. Now, here you will receive capital T in this and capital T in this. Okay, here you will receive small t in this, small t. These are the gametes. Now, if you fuse them, when they fertilize, you will have this. We'll cross these two. We have capital T, these two on this side, and these two on this side. Small t, small t. You will see all the individuals will be capital T and small t. They will receive capital T from one parent and small t from one parent, and this individual will be the offspring. This will be the offspring. Now if you see in the parent one, it has capital T, and means homozygous condition. It was homozygous tall. And this one capital T, small t is another parent. It was again homozygous, but homozygous dwarf. Now this parent, this offspring, capital T, small t, it received one allele from this parent and another allele from this parent, or one allele from male parent and another allele from the female parent. Now if you see, this allele is responsible for the tallness. It makes the plant tall, and small t makes the plant dwarf. Capital T makes it tall and small t makes it dwarf. Now this plant has both the alleles. It has capital T, it has small t. But if you see, this plant is tall. This plant is tall. That means capital T exerted its effects. Its effects were expressed. The phenotype was that of capital T. So this capital T, it is the dominant allele because its phenotype was expressed in the form of tallness, while as small t, which is responsible for the dwarfness of the plant, its phenotype was not expressed. It was not expressed in the offspring because the plant was tall. So the phenotype of small t was not expressed. So it is recessive. This is what law of dominance says. Law of dominance says one allele of the pair of alleles which controls a character usually expresses itself. So out of the two alleles that we receive from the two parents, one from the maternal and one from the paternal side, now these two alleles, out of these two alleles, only one is expressed. Only the phenotype of one plant is expressed, observed in the offspring, and that allele is known as the dominant allele. So here you have capital T is dominant because the plant is tall, so capital T is responsible for the tallness. Its phenotype is expressed, so it is dominant, while as that of small t is not. Small t is responsible for dwarfness. Plant is tall, it's not dwarf, so its phenotype is not expressed, so it is known as recessive allele. Similarly, in this case, we have in cherries we have capital A. So capital A is responsible for the red color of the cherries, and small a is responsible for the yellow color of the cherries. If we cross these two, you have capital A from here in the gametes, you have small a from here in the gametes. When you cross these two, you get an offspring capital A, small a. Here the cherries are red, so the phenotype of capital A is expressed. Capital A is phenotype of capital A is expressed, while as that of small a is not expressed. So capital A is dominant and small a is recessive. There are several other examples that you will get to know as we proceed to other lectures. So in this lecture we studied about the law of paired characters and law of dominance. Both these laws, they are not universally true, they are not universally accepted, and as such these laws are applicable at some instance, while as at some other instance they are not applicable. But the two important laws, the law of segregation and independent assortment, that we will study in lectures, they are the universally acceptable laws, and they are known as the first law and second law of inheritance. They are the fundamental laws that we need to understand, and we'll try to understand them in the next lecture. Thank you very much.